I now call to order the Society's 2,433rd meeting in the 149th year since its founding on March 13th, 1871. Good evening, everyone, and welcome to PSW Science's Spring 2021 meeting and lecture series. Because of COVID-19, the Society is bringing these meetings to you via Zoom from locations all around the globe, rather than our usual home, the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C. This evening's lecture will be about mathematical modeling of epidemic spread and the use of mathematical models to develop effective treatment paradigms and public health measures, with a particular focus on SARS-CoV-2. Our speaker is Allison Hill, assistant professor in the Institute for Computational Medicine at the Johns Hopkins University. I'm Larry Milstein, President and Program Director of PSW Science, one of the oldest scientific societies of Washington, D.C., founded in 1871 as the Philosophical Society of Washington. PSW Science is a 501c3 nonprofit education, science, and professional organization whose mission is to provide a forum to exchange scientific ideas, further scientific understanding, and encourage scientific inquiry. This lecture is being recorded, and it will be posted to the PSW Science YouTube channel, where it will join over 150 other recordings of PSW Science lectures. We invite you all to explore these presentations and to become a member through the Society's website, www.pswscience.org. Membership is a critical foundation of the Society. I encourage everyone with an interest in science and in the Society to become a member. The Society is grateful for the sponsorships of the 2020-2021 lecture series by the Policy Studies Organization in cooperation with the American Public University, and by another generous donor who has asked to remain unnamed. And we are also grateful to the sponsor of tonight's lecture, our very own PSW member, Tim Thomas. We thank all of our sponsors. Before we turn to the lecture tonight, it is the tradition of the Society to welcome new members and to read the minutes of the previous meeting and the summary of the previous meeting's lecture. I am pleased to announce that tonight's speaker, Allison Hill, has been elected to membership in the Society. Allison learned of PSW through our invitation to give tonight's lecture, and her interests will be clear, in part, from tonight's presentation. Welcome to the Society, Allison. Recording Secretary James Heelan will now read the minutes of the 2,432nd meeting and the lecture by Michael Saylor on how nanomaterials allow the development of the vaccines currently being deployed to immunize against SARS-CoV-2. His lecture is available for everyone without charge on the PSW Science YouTube channel, the PSW Science Vimeo channel, and it can be accessed directly from the PSW Science website. James, the screen is yours.
Thank you, Larry. On January 8th, 2021, by Zoom video conference broadcast on the PSW Science YouTube channel, President Larry Milstein called the 2,432nd meeting of the Society to order at 8:02 p.m. Eastern Standard Time. He announced the order of business and welcomed new members. The recording secretary then read the minutes of the previous meeting. President Milstein then introduced the speaker for the evening, Michael J. Saylor, Distinguished Professor of Chemistry and Biochemistry and Director of the Materials Research Science and Engineering Center at the University of California at San Diego. His lecture was titled, "The Year Nanomaterials Saved the World from COVID-19, Nanoparticle-Enabled Delivery of Nucleic Acid Medicines." The COVID-19 vaccines currently approved for use in the United States, developed by Pfizer and Moderna, are both enabled by nanotechnology. Both vaccines utilize messenger RNA, enabled by a lipid nanoparticle delivery system. The forthcoming Novavax vaccine also is enabled by nanoparticles. The anti-cancer drug Doxil was the first FDA-approved nanodrug. Approved in 1995, Doxil used nanoparticles to improve the solubility of doxorubicin and to mitigate its harsh side effects by using the enhanced permeability and retention effect to concentrate the drug in tumors rather than circulating it throughout the body. Doxil showed that nanoparticles could improve the efficacy of existing drugs, rescue failed drugs, and showed the promise of nanoparticles to protect emerging biologic therapeutics. Unlike small molecule drugs, nucleic acids degrade quickly inside the body. To be effective, these therapies need to be protected until they can be delivered directly into the cell. To achieve these objectives, the Pfizer and Moderna COVID-19 vaccines both embed mRNA in a lipid nanoparticle and surround it in a lipid outer shell. Saylor's lab makes nanomaterials from silicon because the body readily excretes it without ill effect. Beginning with silicon wafers, Saylor's lab uses electrochemical etching to produce freestanding porous silicon nanoparticles. Unlike neutral molecules such as doxorubicin, DNA and RNA are highly charged. To coax them together, Saylor's lab first used cationic lipids to ion pair with the nucleic acids to balance their charge. The lab subsequently used calcium chloride to cement nucleic acids into the pores of their silicon nanoparticles, achieving up to 20 times higher mass loading compared to the conventional lipid-based nanoparticles. Unprotected, nucleic acids cemented to nanoparticles would dissolve quickly in the human body. To solve that problem, Saylor's lab wrapped the nanoparticles in a lipid coating. Saylor said these coated nanoparticles should fully release their payloads into the body over an approximately 24-hour period. To target nanoparticle delivery of therapeutics directly into cells, Saylor's lab coats the cationic head groups of lipids around the nanoparticle with polyethylene glycol. This fusogenic coating causes the nanoparticle to fuse with the cell wall rather than being consumed and assaulted by the cell's defenses. The lipid coating then dissolves and the nucleic acids are released directly into the cell. To target the desired cells, Saylor's lab uses peptides with strong affinities for specific cell types to allow nanoparticles to selectively bind inside the body. Saylor then described how his lab employed their techniques to deliver anti-inflammatory therapeutics to treat Staph aureus infections in mice. They cemented a siRNA payload of IRF5 genes to silicon nanoparticles, coated them in fusogenic liposomes, and added peptides to target macrophages that cause fatal inflammation. All control mice died, while all treated mice survived. The speaker then answered questions from the online viewing audience. One member asked whether there is a targeting peptide on the COVID-19 nanoparticles. Saylor said he understood those vaccines do not target specific cells. Another member asked whether the nanoparticle treatment caused any damage to mice livers in the Staph aureus treatment experiment. Saylor said his team looked for and did not find any lesions on the livers of treated mice. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:40 p.m., President Milstein adjourned the meeting. The temperature in Washington, D.C., 2 degrees Celsius. Weather, partly cloudy. The number of concurrent viewers on the Zoom and YouTube live stream, 88, and views on the PSW Science YouTube and Vimeo channels, 156. Respectfully submitted, James Heelan, Recording Secretary.
Thank you, James. Well done. The minutes will be posted to the website in due course. Please send any corrections or comments on the minutes to corresponding secretary Robin Taylor at correspondingsec@pswscience.org. We now turn to tonight's lecture. It's my pleasure to introduce tonight's speaker, Allison Hill, who is joining us from Baltimore, Maryland. Allison is assistant professor in the Institute for Computational Medicine at the Johns Hopkins University. She is also a co-chair of the COVID-19 Data Forum and a co-chair of the COVID-19 Dynamics and Evolution meeting. Allison and our team develop mathematical models and computational tools to understand, predict, and treat infectious diseases, with a particular focus on HIV/AIDS, COVID-19, antiviral immune responses in general, and drug-resistant bacterial infections. Allison is an author on numerous technical articles, and she has written in less technical publications as well, including an excellent article recently published in Physics Today, which I recommend to everyone interested in tonight's subject. Among other honors and awards, Allison received an NIH Director's Early Independence Award and was selected as one of the World Economic Forum's Young Scientists. She earned a BS at Queen's University, Canada, and a PhD through the Harvard MIT Division of Health Sciences and Technology, Medical Engineering, and Medical Physics. All questions will be fielded in a Q&A session after the lecture. Allison, the screen is yours.
Wonderful. Hello, good evening, everyone. Thank you so much, Larry, for the introduction, as well as the invitation to join the society. And thank you to everyone for joining tonight. So let me just get things set up and we'll get started. All right. Great. Okay. All set. So today I'm going to tell you a bit about how we use math to understand and quantify and predict the spread of infectious diseases, with a particular focus on COVID-19. And some of this will cover work that my group is doing, but also I'm talking much more broadly about this field and the type of tools we use and the type of challenges that we deal with. So these days, it's hard to go more than even an hour without being reminded that we're in the midst of a pandemic and that really this tiny virus, less than a micron in size, has changed life as we once knew it. But how can we really understand this? So somewhere in the world, probably not so long ago, a tiny snippet of RNA protected by a protein coat found itself inside the body of a human, a new host that it had never seen before. And just by some crazy chance, this particular virus happened to have some variations in its genetic code. So some mutations that allowed it to enter inside the cells of this new host, hijack its replication machinery, and make thousands of copies of itself. These viral progeny then found other cells to infect, which then produced even more virus, and infection took hold in this host's body. But somehow this virus had some defenses, and so an immune response was probably triggered originally after an infection, and probably amplified as the virus amplified, and this would have set off a whole range of signals in the body. But somehow this virus managed to evade the immune attackers, probably tricking them into some type of self-destructive pathway instead. Then some viral particles managed to hitch a ride with some respiratory droplets that this host expelled, maybe while coughing or sneezing or perhaps just talking. And somehow, against all odds, one of these expelled viruses not only landed at a second host, but again escaped its defenses. This led to the beginning of a cascade of infections, maybe first sort of stuttering along, always on the brink of extinction, but then eventually exploding, taking advantage of the most intimate social networks that we humans have created. Probably first spread in tight-knit groups like families and coworkers, and followed some travelers heading into town for the holidays, and inevitably took advantage of our global transportation network, sparked new clusters of infection in unsuspecting settings. It's triggered dramatic and sometimes unexpected changes in human behavior, sort of cutting off many of its transmission pathways, but it's persisted nonetheless, spreading cryptically in certain subpopulations, kind of hopping around these residual kind of edges in our human contact network. So if we really want to understand COVID-19 and all the other infections that came before it that we're still living with, then we really have to think about all of these processes. Now, infectious diseases are perhaps the best real-life example of a really complex and dynamic system. So think about the type of questions that have been posed in the news over the course of the pandemic. So things like these headlines I have here. So how much do we need to flatten the curve to prevent overflowing our healthcare capacity? Or what would it mean if we tried to achieve herd immunity? How important are super spreaders in transmitting COVID-19? Why would a therapy that suppresses the immune system be helpful for treating this infection? Are colleges testing students frequently enough to prevent outbreaks? How impactful would a partially effective vaccine be? And what if immunity is not permanent? So these type of questions, they can't be answered with careful reasoning alone. And they also aren't really questions that we can generally design experiments to answer. To answer this type of question, we need to have models. So we need to take what we know about all the interacting parts of this complex system and put them in the formal language of math, and then try to understand the emergent dynamics that come out and how we can perturb them. So these are the type of questions that motivate the field of infectious disease modeling, which I work in and which I hope to give you a flavor for today. So actually, similarly to physics, the field of infectious disease dynamics is governed by some high-level principles. And also kind of similar to physics, many of these principles can be understood by considering relatively simple models. So by far, the most common class of models are what we call compartmental models. And these models classify individuals into discrete stages of infection. And then they track the transition of individuals between these states. So in the very simplest model, individuals can be classified by whether they are uninfected and yet susceptible to being infected, or whether they are currently infected, or whether they are recovered and potentially immune to becoming reinfected. And these dynamics can be represented most commonly by a system of differential equations. That's what I've shown here. Or similarly, you could describe the process by some type of sort of stochastic reactions, just like you would in chemistry. These models actually distill a huge amount of variability between individuals that exists in the real world. Like, for example, that my susceptibility to disease might depend on my general health, or that I might be considered sort of more or less infected based on the actual number of viruses in my body at a specific time. But the models can still be very useful. So one emergent property of this system that ends up being very important is that we can kind of distill a lot of the dynamics into one parameter called the basic reproduction ratio, or basic reproduction number, means the same thing. So this quantity describes the average number of secondary infections that are produced by one single infected individual introduced into an otherwise susceptible population. So here, one person infects 3 people, and not each of those people infects 3 others. So that would be a case of R0 equals 3. This quantity has a lot of interesting mathematical properties. It actually represents like a bifurcation in the system. So when R0 is less than 1, the infection is destined to decrease and die out quickly, whereas when R0 is greater than 1, an epidemic can spread. So for example, in this figure here, this blue curve is showing in a simulation the number of infected individuals increasing over time. So this is an example of a case where R0 was greater than 1. And consequently, the number of susceptible individuals go down, the number of recovered individuals is increasing. So R0 can be higher if there are more contacts between individuals, if the per contact probability of transmission is higher, or if the duration of infection is longer. So that would mean like a slower recovery rate. And it's really important to remember that R0 is not really like an intrinsic biological property of a particular microbe. It depends a lot and maybe mostly on human behavior and on environmental factors. So another observation is that early in an outbreak, infections grow exponentially. And this occurs because the rate limiting part of this whole equation, which is the susceptible individuals, haven't really become limited early on. So despite the fact that this is a nonlinear model, you can get exponential growth early on. And this exponential growth is not, the value of it is not determined by R0 alone. Instead, it also depends on the time scale of the infection process. And this time scale is determined by our means, like the typical time between when someone is infected and when they infect secondary cases. So as an example, HIV and the common cold might have similar R0 values, but HIV outbreaks grow much more slowly because the typical HIV infected individual infects a few others over the course of their multi-year disease course, whereas most common colds are very short-lived infections. So this simple model also demonstrates something else, which is that in the absence of any type of intervention, we still don't expect everyone in the population to be infected. So in this example, you can see the epidemic's kind of over, and this yellow curve for the total people who have recovered. In this example, it's around 80%, whereas there's still like some percent of the population that's susceptible. So as the availability of susceptible individuals decreases over the course of the epidemic, the speed of spread is reduced till eventually the spread is subcritical, and then the infection starts to die out. And the time that it takes for an epidemic to run its course and the total percent of the population that will eventually be infected are increasing functions of R0. So finally, even this simple model gives us another really important idea, which is the concept of a vaccination threshold or a herd immunity threshold. So we can reduce the effective R naught, that's this basic reproductive ratio, we call it R naught, of an infection to be below 1 by sort of moving some individuals into this R compartment by vaccination. And the fraction of individuals that need to be protected this way depends on the intrinsic starting R naught value. And this equation tells us, based on the R naught value in this simple example model, how many people we would need to vaccinate. If the vaccine is introduced after some individuals have already naturally recovered and become immune from infection, as opposed to before the outbreak even begins, the fraction that needs to be vaccinated is lower. And in more complex models, this can be a more complex relationship. And note that in the absence of a vaccine, the final size of the epidemic is actually quite a bit larger than this vaccination threshold. So uncontrolled epidemics tend to overshoot this threshold. So just as this example, this simulation that I ran had an R naught of around 2, which would suggest you would only need to vaccinate, maybe 50% of the population to prevent this from spreading. But spreading naturally, it ended up infecting closer to 80%. So it's much better to achieve herd immunity through vaccination versus letting an infection sort of spread naturally. So this model I'm showing here, it's called the SIR model based on the names of these 3 compartments that we decided to divide individuals up into. And it's the most well-known infection model, but it's by far the only one. So it refers to a very particular type of infection in which someone becomes contagious right after they're infected, and in which there's always lifelong immunity, and we ignore demographic processes such as births, deaths, migration. And there are many, many variations of this model that are used to study different types of infections. And we'll discuss some of them shortly in the context of COVID-19. So all of these general principles have some very specific and important applications to the study of COVID-19 over the past year. And you've probably heard many of them being discussed in the news, on Twitter, in the scientific literature, etc. So rapid exponential growth was observed in many places around the world when COVID first emerged. It's been seen again and again during second waves and third waves when control measures were relaxed. And quantifying this exponential growth rate can be useful in and of itself. So for example, for making short-term predictions about the trajectory of cases in the absence of additional controls. And this is still a primary method used in many forecasting applications. Exponential growth rates have been as high as sort of corresponding to doubling times of every 3 days, 4 days, 5 days in some settings, particularly early on in the epidemic. Usually after interventions, these get a lot lower. So, in many places now, even though it's very rapid growth, maybe a doubling time of 2 weeks or something like that. But quantifying this exponential growth is not really that helpful for longer term predictions, because we know that the exponential phase of growth doesn't last forever, and it doesn't tell us how strong interventions need to be. And for that, we really need to estimate R naught, this basic reproduction number. You might think that if we wanted to estimate R naught, the easiest way to do it would be to just measure it directly. So just observing chains of infection, maybe early in an outbreak, we might be able to track everyone who's infected, figure out who they were infected by, and then just count up how many people get infected by each person, take the average. So for example, this image shows a transmission network that was started by a group of Zumba fitness instructors in South Korea. And then let's here, and then to the instructors is purple, and then spread to other sort of students in their class and other people in the general population. So there are other networks like this that we could construct from other well-known outbreaks. But this method can be pretty biased for 2 reasons. So firstly, you're more likely to observe these large transmission clusters, when there's some type of super spreading event, meaning someone infects way more people than on average. And so that might bias our estimates of R naught upwards. Secondly, if the public health system is aware of and tracking infected individuals, they're also probably applying some strong measures to limit onward transmission from these individuals. So you can actually see this in this diagram if you look after a few generations of spread. So for example, by the time you get to these individuals on the edge of this network, the outbreak is stopped. So this can bias estimates of R naught downwards from what they might be in the general population. So there's a lot of potential biases. And for this reason, epidemiologists tend to prefer indirect methods of estimating R naught, meaning we try to back it out from the observed rate of exponential growth that we might see in different settings using some type of model, like we talked about earlier, or mathematical equation. So I said a few slides ago that the translation between this growth rate we see and the underlying R naught depends on this time scale. And in COVID-19, this time scale depends on two quantities. So one is the time interval between when someone is first infected and when they become contagious. So that's called this latent period. And then the second interval is the interval between when someone starts becoming contagious to others and when they are no longer infectious. This is called the infectious period. Expressed in model form, basically, we are considering kind of an augmented version of this model, this SIR model I showed earlier, where we have this extra compartment for individuals who are exposed to the infection, but not yet infectious. And together, these intervals here, they determine this time scale because it's really during this infectious period that this first individual could sort of cause an infection in a second individual. So before estimating R naught, we first have to estimate the duration of these time intervals for any particular infection. And then we need data that actually tracks transmission pairs and where we know who infected whom. So we can kind of estimate the time between when someone might have been infected and when they might have infected someone else and use that to back out these intervals. Then we can translate an observed exponential growth rate into an R naught value. And as an example here, for some particular values of parameters, we could say if we observed a particular doubling time, what R naught value would that correspond to? So for COVID-19, these estimates initially led to estimates of R naught of around 2. Some of them have been sort of adjusted upwards more recently based on better estimates of these intervals. And R naught, of course, depends on the setting. So we've seen different exponential growth rates, different parameters estimated from different settings. All right. So we've talked a lot about parameters related to infection spread and control, but another really important quantity we want to estimate during an outbreak is how deadly it is. So that is what portion of infected individuals, red ones here, will go on to die versus recover. And this quantity is called the infection fatality risk, or IFR, similar to something called CFR, or case fatality risk, that you might have heard about more commonly. And we need this to build useful models to estimate the burden of disease. So one of the biggest mistakes that people made early in the outbreak, if you try to just kind of calculate this back of the envelope, is simply dividing the number of cases by the number of deaths at a particular time. So let me show you here. If we're observing the total number of people who have been infected over time and the total number of deaths that have happened over time, and it's someday we just decide to divide those two numbers and say, oh, that's an estimate of what portion of people die. This green curve here is showing like what estimate you would get there. Whereas this black curve is actually showing, for this particular example, what the true infection fatality risk or case fatality risk is for this disease that I've simulated here. So the reason this method fails is kind of clear when you think about it in terms of model or in terms of the process that's happening here. General, we have quite a big delay between when someone's infected and when they eventually die. And so the deaths that are occurring on any given day are actually sort of just the percentage of cases that happened some days ago. And so for COVID-19, this delay might be around 20 days. And so even if in general, in many populations, this death risk is around 1%, we would estimate something much lower than that initially, even if we observed all cases. Of course, in real life, we have another source of bias, is that sometimes we don't observe all cases and we have to find a way to count for them also. One way that we account for that other problem is that we really need to actually sort of follow a cohort of individuals, not just take a ratio of sort of counts reported for the whole population at a time, but follow a cohort of individuals till they all have an outcome, take a ratio there, and to account for the fact that sometimes cases are missed in sort of just routine diagnostics. Usually we do some type of study where we take a random sample of the population and try to estimate how many people in total have had infection, for example, using antibody-based tests. And so that can allow us to get at a more accurate estimate of this infection fatality risk. So with an understanding of these big picture concepts and kind of good estimates of these key epidemiological parameters for COVID-19, we can build more comprehensive models that can then account for a specific clinical course of a particular infection. And that can then be used to make more specific policy evaluations. So just as an example, one of the main goals of COVID modeling has been to understand the potential strain on healthcare capacity due to the portion of infected individuals who require hospitalization or ICU level care. So many people were trying to estimate this early on in the epidemic. So then we need to make an adjusted model. We know, not everyone recovers from COVID-19. Some individuals die, and some individuals progress to these other stages of severe infection or requiring ICU care, eventual death. So these type of models can be extended to account for all of this. They can be calibrated or parameterized based on existing data for how likely these different outcomes are, and to kind of capture the R0, the exponential growth rate for these infections. And despite all the uncertainties in these parameters, especially early on in the epidemic, the models made it very clear that if the epidemic was allowed to run its course, we would massively overrun our existing healthcare capacity. So here, just recreated some of the common predictions of a sort of unmitigated epidemic versus one that might be more mitigated. So the models can be used to shed light on the potential impact of social distancing measures. So that means sort of strategies to reduce transmission by reducing, in general, contact between people. And these sort of have been called attempts to flatten the curve. And that usually means reducing R0, but not below 1. And another thing that many models showed early on is that that strategy can result in sort of lower peak number of people infected and sort of peak healthcare usage, but it would really still put us at a stage where we're overflowing our healthcare capacity. So that suggested that for COVID-19, we're really in a situation where we can't just sort of flatten the curve, but we really need to crush it, meaning getting R0 below 1 in order to avoid these overflows. Here's an example of these 3 different scenarios here. And these different curves are kind of tracking these different stages of infection, and the dotted lines are showing sort of capacities relevant to these different variables. So for example, projected number of people in ICU versus projected capacity in purple. And you can see that it's really only in this sort of crushing scenario that the projections are that the curve is below the potential capacity. So that was an important finding of models early on and has really helped us decide how we should deal with COVID. So models also give important insight into kind of what we would expect to see after we would put in place this type of intervention, sort of lockdown style. So we expect these interventions to pretty quickly decrease the generation of new infections, but it can actually take weeks for hospitalizations and deaths to peak and then eventually turn around. So here I'm showing some output from a model for, given different intervention efficacies, how long, and so the days after, a really strong social distancing policy was enacted, how long would it take until we see peaks in these different measures of infection burden? And it can be many weeks until we see healthcare burden and the deaths that are occurring peak. So this means really that we have to plan ahead, put the interventions way before we get to the stage of overflowing capacity, and sort of be prepared that it's going to take a while to see the effect, sort of not get discouraged by that. All right. So here's just some example data kind of showing this effect in a lot of different scenarios. These are sort of scenarios from spring 2020 of different cities or regions that implemented really strong interventions after the initial spring wave, and then saw quite a long time until different disease metrics ended up turning around. So for example, here in New York, you can see cases turn around more quickly, but hospitalization, ICU use, and death takes quite a while to turn around. So these models that I've shown so far, showed you how you could adapt this to COVID epidemiology and particular questions you want to answer, and many different models that led up important insights about COVID use these type of ideas. So for example, the type of models that sort of promoted the strong social distancing, models that suggested how contact tracing could be effective, but why we need to do it really quickly since so much transmission happens like right after symptom onset, models that looked into, for example, how frequently we would need to do like random testing in a population like a college, for example, to keep it open during COVID. So all those type of models used really similar frameworks to what I was describing, and just sort of made changes to address the particular question they were interested in. But there's one sort of downside or important assumption to think about with sort of that whole modeling framework that I've shown you so far. And they've all included a pretty big simplification in the way that they model transmission. So transmission occurs when there's contact between an infected and a susceptible individual. And in all these examples I showed so far, it's modeled as what's called a mass action process. So that means that the overall infection rate, this term here, here in the equation, it's proportional to the total number of susceptible individuals and the total number of infected individuals. So that means it's modeled just like a chemical reaction occurring in a well-mixed solution. So I think you can think of lots of reasons why human behavior might not exactly be the same as a chemical reaction. It can be a great approximation a lot of the time, but sometimes we need to go beyond that. So if you think about describing all the possible contacts between individuals in a population as a network, then this assumption is equivalent to assuming what we call a fully connected network. Everyone could possibly connect to anyone else. And it's a valid approximation in many cases, but in the past, maybe 20 years, many people studying infectious disease dynamics realized that this sort of well-mixed population cannot describe the real world a lot of the time. So some examples of why we know that. So there's some cases where we can actually map out a relevant contact network for a particular disease. And during the AIDS epidemic, the importance of sexual contact networks became appreciated. So what this graph is showing is the distribution of the number of sexual partners individuals have. And there's a huge amount of heterogeneity. So most individuals have only one partner, but there's this big tail of the distribution. So this is on a log scale. Some individuals have many partners. And the actual shape of this network is called a scale-free or power law network. It occurs in lots of natural and social systems, and turns out it's important for disease spread. So this second figure shows an actual transmission chain, transmission history of SARS, so the original SARS outbreak in Singapore in 2003. And as you can see, most people spread the disease just to kind of one or two other people, but there are certain individuals, these ones that have numbers by them, who spread the infection to a large number of contacts. And this distribution of secondary infections can't be explained by kind of a random model in which everyone has the same number of contacts or the same potential for transmission. These are just some examples of why human contacts are often better described by a more realistic network that kind of encompasses all the transmission paths that could be possible if infection was introduced into the population. So based on these ideas, it's now common to explicitly consider these contact networks when modeling disease spread. And these scenarios are usually modeled using stochastic processes instead of differential equations. So I'm just showing a little example here for a model called an SIS model, meaning there's no immunity to the disease. You can become infected, and then when you recover, you're just susceptible again. So we use this, for example, for many sexually transmitted infections. And so we do a stochastic model. An individual who is susceptible kind of can interact with an individual who's infected only if there is a contact between them, which we usually describe by some matrix, that's the A here. And in that case, then that individual can become infected. But if there's no contact, there's sort of no edge in the network between them, then they're not interacting and infection's not spreading there. And so in stochastic models, average dynamics are sort of the same as the models we looked at earlier that are just based on these differential equations. But you can see a lot of variation can occur. So just different iterations of the exact same process can lead very different outcomes. And there's other interesting things that happen when we look at disease spread on networks. So one thing we can look at, for example, is the different risks of infection of different individuals depending on the number of contacts they have. And this is just an example from a little simulation, and individuals who had more contacts overall had a higher risk of infection by the end of the outbreak. And it can also be influenced, risk can be influenced by lots of higher order network properties. So over the years, many important principles have been worked out related to the structure of contact networks and how it influences the extent of disease spread. So I'll just run through some of the really important and I think interesting ones here. So in this figure, I'm showing 3 possible idealized transmission networks with different structures. So this one in blue, call it a uniform random network, and every individual in the population is just connected to some number of randomly chosen other individuals, and in this case, sort of 4 or 5 other individuals in the population randomly. Then this green network here, sort of individuals actually have a concept of a position, and then they are preferentially connected to their neighbors. So you can sort of see like people are in a ring here. And then there's some random connections between non-neighbors. And this creates a highly clustered small world network. That's what we call it. So meaning that there's many times if you're connected to someone and you're connected to another person, it's very likely that those 2 people are connected to each other. And this is a property we see in a lot of human contact networks, and it's important to represent. And then this third network that's in pink is very heterogeneous. So the number of contacts that everyone has is chosen from a long-tailed distribution. So this might be more similar to sort of the sexual contact networks, but also we actually see this for many types of human contacts to different extremes. So if you run the same epidemic model with the same parameters, starting from one infected individual in each of these cases, you'll see very different epidemic curves here. So you can see that the epidemic growth is fastest in this pink or very heterogeneous network because it's boosted by highly connected super spreaders. And this spread is slowest in this really kind of clustered small world network where this high degree of interconnectedness actually limits the susceptible contacts that are seen by a typical infected individual. Another outcome that we can look at is the final epidemic size. So for a disease that confers immunity, when it's kind of run its course through the population, what percent of people in the population have ever been infected? And so this showing histograms of the different epidemic sizes. Many times the epidemic just dies at a small size. That's all these times. But if we look at the sort of all the times it didn't die out, you can see that the final size kind of on average is generally highest in this case in the blue uniform network and lowest in this pink heterogeneous network. So this effect actually was the subject of a lot of discussion this summer in the popular press about COVID-19. So the idea was floating around that the herd immunity threshold, so how many people we need to get infected before kind of the population is like roughly immune and infection will start dying out. Some people were claiming it might be much lower than what models had predicted early on, because those models had sort of assumed kind of a more randomly mixing population, and because humans are really structured, maybe the herd immunity threshold would be really low, and maybe we were already there. So you can see, that effect qualitatively is true. The threshold is a little bit lower in these heterogeneous networks. It happens because highly connected individuals are more likely to get infected early on. Then they become immune, and they kind of act like immune hubs, like blocking infection spread around the rest of the population. But contrary to a lot of reports, the threshold and sort of the final size of the epidemic is only a little bit lower when you end up putting realistic parameters in. You often don't get like a huge difference in the final size. And if you simulated things a little differently and you actually consider that this network might not be fixed, so we might be someone who has a lot of connections one day or 1 month might have less another month, and someone else has a lot of contacts, then a lot of these effects actually disappear. So they're less robust than it might seem. So we became interested in how one really important aspect of transmission network structure impacts COVID-19 spread and also control. So we think about the main tool that has been used to control COVID around the world. It's some sort of social distancing, lockdown, stay-at-home orders, etc. And to a different degree, these sorts of blunt policies are what they're doing is reducing the number of potentially infectious contacts people have, but only outside their household. So people have contact inside their household and outside, and none of these policies are really doing anything to reduce infection within a household, sort of only spread between households. And we know that households are a really important contributor to COVID transmission and for other respiratory infections. And many of these policies maybe even increase the amount of time that people spend at home with their household members. So how does this affect epidemic dynamics and our predictions about how successful certain measures might be? So to get at this question, we created transmission networks that include connections within the household and outside the household. And household sizes can be extracted from census data, and you can assume roughly all household members can contact all other household members. And other types of surveys and data can help us estimate the distribution of contacts outside the household that people have. And with this network, you can then simulate a COVID model, sort of like ones I've shown earlier, implement social distancing measures, which is equivalent to removing some of these contacts, and then see sort of what you expect. And one thing is that we don't actually know precisely the contribution that each type of contact plays to transmission. Like now our estimates of this are getting a little more refined, but there's still a lot of uncertainty. So household contacts, for example, could have a higher transmission rate than an external contact. So you might have more intense physical contact with people in your family, for example. But with external contacts, maybe you actually just spend more waking time of the day if it's the contact at work or at school, and that could lead to sort of given that contact a higher chance that you might spread infection to them. And so because studies have found a range of values here, to answer this question, you kind of want to consider a range of different parameters, not just focus on one, which is something that comes up a lot when we do infection modeling. So, for example, what I'm showing here, you consider that these two types of contacts might have the same weight, or that the contacts outside the house might have a lower weight, or maybe even a higher weight. And here, weight is just meaning that given that the contact exists, and given if one person was infected, how likely that they might infect the other person in a typical time frame. So we found that actually considering these dynamics, sort of produces some pretty interesting and surprising results about the potential efficacy of interventions to control COVID. So firstly, we found that kind of with these realistically structured populations, you need really strong reductions in these external contacts to stop spread, kind of more than you would predict if you didn't consider this network. And that if you have a kind of imperfect reduction of these external contacts, which is the case we're always in, you can have a really sort of slow decline in cases. So for example, this is an example of a pretty effective intervention. You get rid of 80% of external contacts, and you still have a very slow decrease in cases that's predicted. And this is something we've seen in a lot of different settings. Can sort of look at this in a little more depth. So it turns out that the relative importance of these two types of contacts is really important. So in these graphs, this y-axis is a prediction of kind of the final epidemic size after it sort of runs free for a bit and then is controlled later. So it's the percent of people that would have ever been infected at some point under either a fully effective or partially effective socially distancing intervention. And for different sort of contributions of these different types of contacts. And interestingly, models show why social distancing measures are actually most effective, meaning you have the smallest epidemic size prediction when external contacts either contribute a lot to transmission or when they contribute a little bit to transmission. The worst case is the intermediate regime. So why is that? So when external contacts contribute a lot to transmission, then eliminating them eliminates most transmission. So that's a good intervention. When they contribute very little to transmission, even a weak intervention sort of can remove the ability for infection to kind of jump between households. But when you're in this intermediate regime, sort of enough transmission between households to sort of continually seed new households and infection can kind of effectively spread even after the innovation, sort of both within and between households. So like I had mentioned that what if another impact of certain lockdown measures is increasing the time spent at home and increasing the amount of transmission. Of course, that sort of further increases the potential amount of epidemic that could happen even under these measures. So overall, this type of model helps us understand why, because if we're not controlling household spread, we would need to do an even better job of controlling spread outside the house, and why it's really important to design studies early on to try to estimate what are these different sources of transmission, how much are they contributing, so we can try to understand better what strength of interventions we need. There's another effect of this type of network structure, which is that it can lead to even longer delays between, even if you do implement a pretty strong social distancing intervention, how long it takes for the epidemic peak to show up and then turn around. So these different lines on here showing some different colors are different measures of the epidemic burden in terms of sort of all infections or infections that need hospitalization or more critical care or the number of deaths observed. And whenever we have this network structure taken into account, which are the darker colors as compared to the lighter colors, then these delays here, so the time until the infection peaks, are even longer. And the reason is that you could sort of get continual generations of infection within households even after the intervention is going. So that's important really to kind of get our expectations right about what interventions can and can't achieve. So far, the way we've been modeling, the way I was describing kind of modeling social distancing interventions, one way of doing it you could think of as just kind of randomly removing some amount of contacts that people would have had at baseline. And that's sort of maybe the simplest and most common way to model this type of thing. But of course, like everything in the real world, it's more complicated. These outside household contacts, so what we call external contacts, in real life, they can be divided into different types. And we can think of these as like layers of a network. So for example, you can have contacts, workplace, social contacts, school, just general random people you might run into. And a lot of data shows that some of these important sites of contacts, so social groups and the workplace, contacts are highly clustered, meaning that you share friends or contacts with many people. Your contacts share contacts with each other. So if you want to really capture what effect that has on control, you need to think about it a little more carefully. So what this means is that the degree of adoption of any sort of distancing measure that's in place might also be highly clustered. So if you think about it this way, certain occupations might not be able to work from home. So you might have to think about sort of entire workplaces that still exist or others, that might be kind of dissolved. So we can simulate this by actually simulating these different parts of the network and using different parameters to get at the size of these groups and their clustering. And when you do this, you kind of look at the same thing, whether you just modeled it this simple, random removal of contacts versus this more clustered removal. And you see that that can also make a difference for your predictions. So some things look very similar, maybe the size of the peak and when it happens. But what we found is that when you kind of take this into account, this sort of like clustered adoption of social distancing, it can result in much longer times for disease elimination. So here versus here. So basically you can have kind of pockets of the network where infection can persist for long times despite interventions. And these type of findings suggest that targeting demographic groups, essential workers or other groups where this type of thing can be going on, with sort of more aggressive case-based measures, contact tracing, et cetera, be necessary to reach elimination goals faster because of this clustering. So you can use these type of models to understand how an individual's risk of infection, for example, might depend on their household size and make predictions about that. In these type of models, actually predict a greater than linear increase in infection risk versus household size, because actually larger households, there's both more risk of seeding infection from the inside and also more risk of it spreading, sort of more individuals spread to within the household. So that makes some predictions for why larger household size are very risky for infection. And there's lots of examples from around the world where areas where household sizes tend to be larger or more crowded, having worse outbreaks. Could also look, for example, at the increased risk faced by households that have essential workers, meaning more contacts outside the household versus those that don't. And for a particular parameterization, get out some estimates for how much someone's risk is increased. For example, if there's no workers in their house versus living with the worker versus outside. So these are type of predictions a model could make and then can be compared to data. So different studies now are kind of getting at these measures, real outbreak that can be used to kind of refine and make models better for making future predictions. Okay, that's that. So at different times in the epidemic, some regions have encouraged kind of partial relaxation strategies, which is when groups of households sort of merge to form circles or bubbles. They've been sort of called sort of social bubbles or different things in different contexts, but still kind of minimize external contacts. So this is kind of like a merging of households. Example, this could help with childcare, social isolation. And we studied this in our model to try to understand when are sort of forming these household bubbles safe and when might it not be safe. And we found a few important criteria. So one is that these type of sort of bubble formations should really only be recommended or applied in the context of steadily declining cases, deaths. So that's this example here, for example. In situations where in our models, when infection levels had kind of stabilized but weren't really declining, forming bubbles, if a large percent of the population does it, could lead to some resurgence in cases. And we also looked at sort of an extra intervention that could go along with this to find out that these bubble formations should ideally be accompanied by further decrease in external contacts in order to make it more robust to the stage of the epidemic that it's applied. So, for example, saying, there's only one grocery trip per 2-family household versus one trip for each household, even if you're socializing. This can make it more likely to work. And instead, you might just get epidemic stays persistent as opposed to resurging. But it can also help it sort of still go towards elimination despite forming social bubbles. So overall, this is suggesting that policies like this should be thought carefully and only encouraged in regions where certain other conditions are met about the epidemic trajectory. So studying changes in household structure has another really important application to the pandemic, especially in the US. So since spring 2020, unemployment rates have soared and many US households are very behind on paying rent and are at imminent risk of being evicted. And if you're interested in the eviction crisis in general in the US, this is a really great book. Even in the absence of the pandemic, the US has sort of abnormally high sort of rates of people being evicted every year, or at risk of eviction, on the verge of being evicted, and this has become a lot worse with COVID. So it's hard to estimate exactly how many households are at risk. Different estimates suggest that across the country, anywhere from maybe 0.5% to 5% or more of households could face eviction on any given month under the pandemic, under current situations. I mean, if you just look at unemployment rates and how much they've increased and use that to scale up eviction rates, in some cities, you can get very scary estimates. Some are shown here for just some example cities in the US, but a lot of uncertainty under all of this. Other than the not uncertain part is that this is definitely sort of impending disaster. So there was some kind of patchwork temporary legislation preventing evictions in the spring and summer, but all of those policies were set to expire near the end of the summer. And at this time, collaborators and I became very worried about the effects that this might have on COVID transmission, given what we know about household spread. And we wanted to use the model to quantify how important these bans were and how important it was to keep them going. Luckily, in September, the CDC enacted another temporary ban. It has been challenged many times, or quite a few times in federal court, has managed to withstand it so far. And actually, just as of today or yesterday, after I made this slide, this eviction ban has been extended for a few more months. So, is this relevant to COVID? How relevant is this? The whole point, this ban is sort of implemented under a public health act that says that it's supposed to be for measures that are important for reducing disease transmission. And how does that relate to evictions? So what happens when people get evicted? How do we model that? There are three main things that can happen. Someone could find another house to live in. A family could become homeless and have to move to a shelter or to the streets. Or what it turns out is the most common thing that happens is that households tend to double up with other households, meaning one family or one household moves into an existing house, other individuals sort of augmenting the number of people living in one living space. So this can be modeled as we were sort of modeling these social bubbles before, except now eviction is obviously kind of a non-voluntary process. We can model that as two households coming together and increasing the number of individuals sort of in this new household, and therefore causing an increase in contacts. And we can estimate how big that increase in contacts is and how much it might impact the risk for those people, but also for the population at large using these type of models. So I'm just showing some example results here, looking at evictions occurring in the context of an epidemic pattern that has been seen in some major US cities, Philadelphia, Boston, New York, New Orleans, many on the West Coast too, where you saw a really big wave in the spring, sort of some leveling off over the summer, and then this resurgence in the fall. So we considered a scenario where there was or was not this moratorium on evictions happening starting on September 1st. And we looked at the difference between when it was in place and when it wasn't in place in terms of the number of evictions that happened, the number of infections that happened over that time period, given sort of different proportions of households that were subject to eviction and subsequently doubling up. And we found that evictions resulted really in measurable increases in the epidemic size, even when they were relatively rare, leading to potentially thousands of excess infections in a city of around 1 million. So here are some examples here for different eviction rates. This is percent of households that could experience eviction each month, and how much that could cause increase in the epidemic as predicted by this type of model, filled in parameters with like our best knowledge for all these different rates for COVID. And evictions, according to predictions, substantially increase the risk of infection for individuals who experience them. That's these orange lines here, but also increase infection risks for the unaffected individuals due to spillover. So more infection that happens within one household leads to more chance that infection will spread to other households. And so increase the risk really for everyone in the population. And we found that even if we modeled the situation in which infection sort of was controlled starting towards the end of 2020, which was occurring in some places now, evictions happening kind of during this time still increased cases, and they compromised the efficacy of these interventions because they increase the amount of household transmission that is possible, and that is bad for everyone. Those simulations I had showed you kind of assumed evictions, are equally likely to happen, to any random person in a population. Of course, in reality, they might be clustered in lower socioeconomic areas of a city. For example, maybe they're individuals who are disproportionately unable to adhere to social distancing interventions that might all be more likely to be contacting each other and in a particular neighborhood. So you could sort of model that as saying, well, there are sort of two different communities here, one that experiences eviction, one that's less likely to, and they have most contact with each other, but perhaps less contact between them. And you might think, oh, this means that it's really only sort of this population that is affected by evictions and everyone else is fine. But actually, when you model this, find that even when you have evictions clustered in certain demographic groups, and those demographic groups are the ones less able to social distance, the overall impact of the evictions is still quite bad, and it's even worse than if they happen randomly, again, because it can lead to these subpopulations where a lot of infection is transmitted. So again, preventing these type of increased transmission risks really benefits everyone in a city. And these type of models can be made much more detailed. This example of a case study we did to really track what's going on in the city of Philadelphia, really take into account all the different regions of Philadelphia and the different demographics that are there, the different risks of eviction, the different kind of ability to socially distance, which you can measure from a lot of kind of big mobile phone type data. and allows you to kind of put a more fine-tune a model for a particular setting. And then also estimate kind of the risks that can be averted for different groups by preventing evictions from happening. So just sort of one final part of this story is that there's a lot more complex ways that the structure of human contact networks can impact the spread of COVID and many other diseases. These are just some examples that I'm telling you about today. So for example, our collaborators at Northeastern University and Oxford University actually looked in detail across many, well, really all Chinese cities and their outbreaks during winter of 2020. And they looked at many determinants of what determined the different shape of these trajectories that happened in different cities. And they found that the degree of crowding that occurred in those cities, so that's related to a population density factor, was associated with the epidemic path that was observed. And they kind of defined this as saying, oh, there's epidemics could have higher intensity, meaning they were more clustered around a peak versus being more diffuse and spread out. And they can sort of quantify that and like compare it across different cities. And they found something interesting. So they found that epidemics were more intense, meaning that they were more peaked in less crowded cities, so more sparse cities, and that epidemics were a little flatter in more crowded cities. Overall, this is about the shape, not the total number of cases. Crowded cities still had overall a little more cases, but it tended to be more spread out. This is not so easy to explain because if you used a really simple model and you just said that in a crowded city, R naught is higher, each person infects on average more people. Actually, if you simulate that, that leads to a sharper curve, so more intense in crowded cities. So how do you explain that? So we worked with them to try to understand how more details about how human populations are actually structured could explain this. So if you kind of model contacts that occur at different levels of organization. So example, you have households, you have neighborhoods, you have a whole city, and there's sort of more contacts at smaller levels and less at higher levels. This can actually explain some of these findings. So you can actually get more when you have more connections sort of between these different neighborhoods, which can happen more in more crowded cities. That can lead to kind of more jumping around between different neighborhoods, and that can actually spread out an epidemic more as opposed to keeping it kind of all happening more in time. So it's a complicated effect, but it can explain why sometimes all of these levels of human interactions can be important for understanding a pattern that's seen in big data sets and is quite counterintuitive and not explained by a simple model. So I hope that gave you sort of a bit of a flavor for what we think about when we're modeling infections, how we can make models simple to learn some important principles, or how we can add more details to try to get at more nuances of what might be happening for a particular infection, and why this human behavioral aspect of it, because everything I've talked about contacts is of course part of human behavior, is so important for this. And as you know, really being able to estimate numbers and patterns and changes related to human behavior is a lot more difficult than, for example, just measuring sort of a medical or purely biological quantity. So everything that I've talked about today that was sort of contributed by our group was done in some way by some people on this team. So many of the results from us were done by graduate students of mine and at University of Pennsylvania, working under my collaborator, Mike Levy. Some former students from our group kind of came back to help with COVID modeling. We work closely with a group at Urbana-Champaign that works on housing policy and how that intersects with COVID. And then a big team at Northeastern University that helps us think about a lot of these and measure a lot of these network structure human contact patterns and how they impact disease spread. I've included some additional readings here. I know you'll all have access to the videos. You can see it there in terms of sort of more basic learning about how diseases are modeled, how this has been applied to other diseases, as well as some of the references for some of the results that I showed today, which are also, every slide should have references for all the other people's work that I presented. And I'm happy to take any questions that you have. Thanks so much for your attention.
Well, thank you, Allison, for a very, very informative lecture and for providing all the additional material for us to follow up. We have time now for the Q&A session. Zoom participants, you can raise your hand or type your question in the Zoom Q&A panel. I will call on Zoom participants in turn to unmute their mic and ask their question. If you're a Zoom participant, you want to ask a question, but you don't want to do it using your microphone, you can indicate that when you write your question in the text. YouTube viewers, you can text questions in the YouTube chat box. I will read all questions aloud for everyone to hear, and so they show up on the video. So we have a question from Carl Merrill. Carl, if you will unmute your mic, please ask your question.
Hi, thank you. That was a really nice lecture, and it represents a lot of work, I know. But I'm concerned about one aspect of it, and that is that the whole modeling is predicated on a fixed virus. That is a virus with fixed properties, and not one that's mutating. And I know that adds tremendous complication, but in the real world, it becomes a major problem. And in fact, yesterday in the New Yorker magazine online, there were a couple of articles, one where the British are very worried, but another one that argued that with 3 separate mutations, you end up with an escape virus that will not be stopped by any of the antibodies in survivors of the infection. And that would have a profound effect if that occurred in the population, but the model doesn't take that into account. I know that would make it very, very complicated, but I wondered if you could comment on that.
Yeah, so actually my day job is modeling virus evolution before COVID came along. So I study HIV, which is a highly adaptable, mutable virus with a tremendous amount of global diversity that rapidly develops resistance to antibodies and drugs within each person it infects, and also study other drug-resistant infections. So yeah, this is something I think about a lot. And it's true for most COVID modeling, this was really not the thing people were focusing on, because for various reasons, people really downplayed the risk of sort of adaptation of new strains of COVID. And, there's a few reasons. Maybe it was reasonable to think that. Certainly, when you have really short infections like COVID within a person, there's just less time for new variants to arise within one person and then spread at the population. The virus does have sort of a little bit lower sort of mistake rate when it copies its genome in a cell. And in general, like from other coronaviruses that are circulating in humans, there's just less kind of genetic variation compared to some of these infections. So, those were reasons why maybe it wasn't thought about as being the most important thing. But of course, now we're at a situation where hundreds of millions of people around the world have had this infection. And, within every person who's infected, there's billions of viral particles. And so there's just tremendous opportunity for new variants to arise. And because we're now in a situation where we have a significant portion of the population that's already immune, there's a huge selection pressure for new variants to arise. So, it's not surprising we're in this situation. So, what is, to your question, like, how do we model that? So, there's some parts of evolution that are easier harder to model. I mean, it's very difficult to predict sort of what strains will be possible to emerge. And what their phenotype will be in terms of like, will they escape an antibody or will they be more transmissible or more deadly? That's very difficult really to say. You can consider scenarios in terms of if a strain with these properties came up, how likely would it be to take over in a local area, in a global area? How long would that take? How would it sort of change our estimates of burden? So we can consider you can consider those things in models. Basically, what it comes down to is just having sort of multiple different virus species in the model, and then you have to sort of know something about or make some assumptions about how that virus changes all of the parameters in your model and how it might interact with the original strain, like how much cross immunity is there. So
well, the other problem is that the virus, the immunity, how long we stay immune is not known at this point. And so, in fact, the virus presumably, and in fact, we have cases where it comes back, and we don't know how much immunity we'll get from a vaccine versus even the infection, because people are getting reinfected. And as you pointed out, the more people that get infected, the greater the pool is for the mutations to occur. And so if it becomes even more spreadable, that is, it's more infectious, the RO goes up, we'll end up with an epidemic that continues to get worse and worse. That's my own personal concern. And I just wondered if
that's a big concern. I mean, this is another reason, if you didn't have another 100 reasons for this already, why the idea of like, oh, we will achieve herd immunity just by letting the infection spread is a terrible idea, because every time someone's vaccinated, that's like removing, one opportunity for the virus to like generate a bunch of mutations in one person. But when you have infection spreading between this many people, there's so many opportunities. So this is a big problem. It's very scary for natural immunity. It's very scary for vaccine immunity. It's a bad situation.
And I guess one last comment that I'd like to make, and maybe you could comment on it, is that with the first generation of vaccines, one of the things they did not monitor for was infection and shedding of the virus. So you might have a vaccine that cuts the mortality and the morbidity, but may not stop the infection and the shedding of it. And I think people are beginning to just look at that now. And in some ways, that would be the worst scenario, that they wouldn't even know they had any symptoms and continue to spread it.
Yeah, I mean, it's not like, this is something people have modeled, like how good could a transmission blocking vaccine be versus a just severity blocking vaccine versus both. And like, it's still, in general, better to have that vaccine than to have none, as long as people are aware, but it's not as good, obviously, as having one that really stops shedding and transmission. And it's true that hasn't been reported sort of as a outcome and a success of the existing trials. And it is being studied now. I would say that in the animal studies of some of these vaccines, one particularly, like there's a group at Harvard that I know and have worked with at other things who are involved in some vaccine development. And in their trials, vaccines did actually, basically like prevent virus from growing and replicating to high levels in the animals who were vaccinated. So I think, there's a good chance that could be an outcome of the vaccines in humans, but it's true, we don't know, and I hope we know soon.
Thank you.
We have a question from Charles Clark. Charles?
Yes, I'm an atomic physicist, and I have my hands full modeling the interactions of dilute systems of Oswald atoms. And so I'd just like to say one word: respect. The work of you and your colleagues in integrating all these issues of medical and epidemiological and social science in a time of a really great crisis in the world is really amazing. I'm wondering if there's enough information present at hand for you to make a comment on the progress of vaccination. I know it's early days, but there are issues of take-up. For example, many people don't take the flu vaccine in a given year. And do you have any comment on, well, maybe you could just say, do you have a plan for understanding how on a statistical basis, the progress of vaccination can be monitored? And I won't ask another question.
Yeah, I think there's like many aspects to that. I mean, people have been doing kind of recurring surveys about attitudes towards vaccination. And, there was a time when, it sort of seemed like, oh, many people would be very hesitant about the vaccine. That has been sort of substantially improving over time. So as people see like the enthusiasm for healthcare workers for taking the vaccine, as people see that this epidemic is continually getting worse, as they see the sort of lack of severe adverse effects in the millions of people who have taken it, like people, views are evolving. So I think that's a good thing. Will we ever get 100% of people begging for the vaccine? Maybe not. And good thing about all of this is like we don't need that, right? As long as we get a substantial portion of the population, that can potentially be enough. Like one interesting thing that modeling tells us about this is that those like vaccination thresholds that I showed at the very beginning, they can be compromised if all the people who don't take a vaccine tend to be more interacting with each other versus others in the population. So you can sort of get like clusters of disease persistence when otherwise it would be eliminated. Of course, that's what's going on for measles in many settings. So yeah, this is complicated. There's some really nice modeling work on this. It's not something I'm particularly working on, but there's 2 papers in Science. One just came out really recently that a group like kind of jointly between Harvard and Colorado, considering a lot of different scenarios about vaccine uptake. And there's another one from a few months ago by Brian Grenfell's group at Princeton that considers different scenarios in terms of how efficacious the vaccine is and how much it wanes. And so can get some more ideas there.
Have a question from, or a couple of questions from Frederica de Rema. Frederica?
Hi, Alison. Great work. And I myself have led a number of programs on epidemics. And in fact, I would like to connect you and with some other people in the US and in overseas, because there's common work and hopefully you have possible collaborations also. So I have 2 questions, but let me ask the second one. The first one was regarding correlations, and you talked in several contexts, but I can ask the question afterwards if there is time. However, I had also the question, we talk about if there are any studies, because we talk about 1 million vaccines per day for 100 days, 100 million for 100 days. And that's a nice slogan. But are there any kind of studies that whether we should be doing more, we need to do more because of the reasons we discussed in terms of mutations, and the more people get infected, the more the probability of mutations with other effects that we discussed? Or also, can we do less? And right now, the kind of the staging is based on age and pre-existing conditions and so on. However, based on the things that you also kind of presented, should it be in a sense prioritized in terms of communities that have more kind of chances of being infected? Because, for example, service workers and typically culturally and financially such communities also are overcrowded in terms of families and cross families. So have there any studies being made with respect to these aspects?
Yeah, absolutely. Thanks for the question. And yeah, I would love to connect with the colleagues you mentioned. So this is one example of where modeling versus sort of policy are not really meeting. So there's like so much amazing modeling work showing how you can get more out of vaccines by targeting them, in lots of different ways. It could be related to age groups or, hubs in a network, or all of this, depending on what you're trying to optimize. I mean, there was a lot of literature on this before COVID, and there's been nice work on this for COVID. So, I mean, this Science paper from the Harvard and Colorado group, I think, addresses some of these, and there should be forthcoming some other papers about vaccine allocation, both from groups in the US and also Europe, Asia, and they look at this in models. And yes, it can be much better to target groups. The problem is it's just very difficult to implement. I mean, logistically, it's difficult. Ethically, it's difficult. And, we are like having a very difficult time even doing very simple vaccine rules where we just say everyone like over this age or everyone with this job. Like it's, I've heard a lot of disaster stories about that. So it just seems like it's not like we have the answers from models. Like this is now like an implementation, policy, leadership type of problem that's really limiting this from being realized.
Okay, thank you.
We have a question from Dick Garwin.
Yes, thank you. In the graphs reporting 80% intervention and 90% intervention, there are vertical lines at 50 days and at 100 days. What's happening at 50 days and what further at 100 days?
Oh yeah, I don't know exactly what graph you're talking about, but if there's vertical lines, usually we're just sort of simulating some scenarios in terms of the days since the epidemic was started, at what time, assuming that it's like a 1 day change point that a particular sort of policy is introduced.
No. Something happened on the same curve at 50 days and then again at 100 days.
Yeah, so that's probably changes in the amount of contacts people were happening at those points. So
okay, it isn't clear. Thank you.
Yeah, thanks for pointing that out. So usually in order to recreate the path of the epidemic in many US cities and states, and this agrees with what we see from lots of mobility data, there were really dramatic reductions in contacts that happened near the end of March, and those were sustained for some time. And then starting in the summer, contact rates increased again. So in a model, we're marking as just a particular kind of days that those changes are happening.
We'll go to Tim Thomas, our this evening's sponsor, if he can speak. If not, I'll read his question. Tim?
I will speak.
Excellent. Yay.
Yay. Well, thank you for a lovely, interesting lecture and extremely timely. I'm wondering about the impact of improving hospitalizations on case fatalities. I noticed that hospitalizations are going up, but deaths are not at the same rate in some of our jurisdictions around here. And I'm wondering if there are some hospital systems which have dramatically improved results when compared to others.
Yeah, it's a good question. I don't know the answer to that. I think, there's only so much you can do once someone develops like very severe COVID. We don't really have specific treatments. We have sort of, most relatively non-specific things that have certainly been improved over the course of the pandemic. But, a lot of these things are based on protocols. I'm not sure how much they would differ between centers. One possible thing to think about when interpreting that type of data is that people often go to hospitals like in jurisdictions other than where they live. And so that's the hospitalizations are reported based on where the hospital is usually, but deaths are reported based on where the person resides. So sometimes you can get weird things like that happening in the data, but it's hard for me to say like for a specific situation.
You mean the deaths are reported by the coroner? And even if I were to die in Montgomery County, my coroner in Fairfax County would report my death?
No, your death is reported based on where you live.
Not
no
where I die.
So when you die, it is reported as a death in the zip code county, like where you live. This is my understanding of it. Whereas if hospitalizations are tracked based on the hospital where they are occurring. So, we usually don't look at hospitalizations at like a zip code level or even really a county level because people go to hospitals that are not like right beside their houses. There's only so many hospitals. Most people for COVID would go to a major medical center. So that can just cause some discrepancies in like the location where hospitalizations versus deaths are reported.
Okay.
But Tim, wasn't your question that hospitalizations are not down overall, but deaths are. So it's the ratio of deaths to hospitalizations. Is that what you were asking about?
Yeah, that's what I was asking.
But I'm
about.
saying within one particular area, if it's a small area, that could be because of people
Yeah.
being moved to hospitals in different areas. I mean, I don't know. These are just, I can't say without looking at the
Okay,
data.
yeah.
There's possibilities like that.
So
Okay.
let's take a voice question from Bob Terry, and then we'll take one from Phil Nicholson. So Bob?
So do you hear me okay?
Yes. Hi, Bob.
Okay, fine. Fine tour de force, Allison. And very enjoyable presentation. But I'm curious in the sense that COVID patients have a higher and lower infectivity rate as their disease progresses. Sometimes they're infectious even before symptoms, as you have said in some of your binning. So if you have a function which is a strong parameter based on the history of a patient, it seems like if you're going to write a differential equation, you're looking at a time delay operator or convolution over the population. So every patient coming out there in your space has a clock, and you have to have the rate of you getting infected by that person depends on their clock. So you have a convolution back in history to get the infection rate at any given time. And so I was curious. Yeah, with a whiteboard I can show you, but I'm just trying
I know
to
what you're
describe.
saying.
Yeah.
Yeah.
Now, has that been applied, and what has been the result of looking at those sort of things? 'Cause I think it's an interesting system.
Mhm.
I'm just curious about, is it worth doing?
Yeah, absolutely. So for particular modeling questions, that's really important to take that into account that way. And there are multiple papers that do that, including some really high profile ones. And so, like I said, all of these models are making some assumptions. And one of the assumptions, obviously, in the one I was showing you, is just dividing up those intervals into discrete time. So instead of like a continuous level of virus, we say, okay, you're not infectious. This is the time period when virus is high enough. You are infectious. And this is the time period where, you're no longer infectious. But you could make that a continuum. And it's important sometimes for studies that are asking about how quickly do you need to isolate someone? How quickly do you need to conduct contact tracing? How much does that reduce sort of the number of infectious people out there? It was included in some of these studies about sort of closed populations where you're doing really frequent random screening, like the college sort of situation. They might have included those details. It's important if you wanted to kind of make a prediction about kind of for someone like, let's say, a returning traveler, and instead of quarantining, you want to like just get tested 2 or 3 times. What are the times to do that? Like, then you would probably want to take that whole trajectory into account. And yeah, you can do that the way that you're describing. You can also do it other ways. So for example, by dividing these states I've shown just into smaller categories. So like each different day of infection instead of like a week of infection is a different category. So sort of discretize viral load. That's an example. Or you can make a full model that models what happens in one person's body and the whole virus is growing, virus is getting cleared, and like a multi-level model. So there's a lot of ways to do that.
I was just suggesting also that if you have these networks as you see them, and you look at the topology and the connectivity of the networks, it seems to me that if you look over a fairly broad swath of possible networks, you can map that back onto a normal standard infectivity rate. You just have to be intelligent about how much variation you might see in that rate to span the network space.
Yeah, it's not actually mathematically equivalent. Some properties are captured by doing that, but having what you're basically describing is kind of equivalent to having like how to explain this, like a mixing population, but with heterogeneous sort of amounts. Imagine if you have a lot of like chemicals swimming around in a swarm, but each of them have different radiuses that like when someone gets in there, they can have a reaction, which is not the same as having like a fixed network of what could happen or not, because it actually just changes the infection dynamics in some way. But it's sort of a conversation, like an interesting math conversation for another day.
Well, that's what I would suggest. I would like to make contact with some of your colleagues and show you some of these modeling ideas and get some discussion. Can I get an email or get some papers from you, Larry?
Yeah,
Is that going to be there? That contact information will be possible.
I am easily Googleable, unfortunately.
Well, I won't bother you very much, but
No,
thank you.
I'm just joking. I'm happy.
Thank you. Thank you for the invitation. All right. Thank you much.
Thanks.
Phil Nicholson, astronomer from Cornell. Phil?
Thank you, Larry. Thank you, Allison, for an excellent talk. Even though I am an astronomer and not a biologist, most of it made sense. At least the differential equations made sense. In the context of your sort of household models, I was reminded of something that there's been a fair amount of discussion here in New York State as to the priority that should be given to vaccinating prison populations, which is a rather peculiar household case where there's obviously a lot of intimate contact between hundreds of people in a prison, but limited contact with the outside world, except of course through the prison guards who go to and from every day. So there's been arguments that they should be really a high priority in the context of your targeted vaccinations. I think unfortunately that has really not happened because they don't have many political supporters in the legislature. But I wondered if you had done any modeling or you knew of people that had in that direction.
Yeah, it's a great question. I sort of gave some advice to some groups that I think from Yale who were doing some modeling on this, and I've seen that there is 1 or 2 published papers related to this. Yeah, your intuitions are correct. Obviously prison is like a giant household and there's really high risk of spread there. And it's not we can't just, even if you didn't care for prisoners, and you were just saying, oh, well, it just doesn't matter what happens there. They're separated from us. As you stated, that's not even true because there is, there's employees and contractors and everyone going back and forth. So, it's important for everyone. And yeah, people have talked about prioritizing them for vaccine. And this is just another example. Obviously, that's what the epidemiology and the modeling would suggest would be a priority, but this is just not a political bestseller, unfortunately. And, there are definitely many public health people who continue to advocate for this and point it out, but there's only so much sway that they have.
Okay, thank you.
Okay. Take a question from the web, and then we'll go to Peter Kessler, and then we'll go to Charles Norton, just so they know they're going to be up soon. From the web, via our social media director, Ann McQueen, what is the most common or most harmful mistake that you've seen with people getting into doing COVID modeling?
Yeah, that's a good question. I mean, overall, I would say it's great that people have gotten into COVID modeling. I mean, it's great to have more hands on deck.
That was from Will Angel. Sorry.
Oh, okay, no problem. But yeah, certainly this is always a concern when you have people jumping into it who maybe really love to do the math, but are really not thinking about all of the biology and medical and sort of epidemiological and behavioral aspects of this, and thinking that they're the first person that ever thought about making a network. I think a mistake that happens a lot is people thinking that more complexity to adding that to a model is better, and that's not always true. The more free parameters you have, the less idea you have what's going on and why, and that can make your predictions very unstable. So the reason people prefer simple models is you really understand what is driving the dynamics. But I guess one of the worst outcomes is these groups that have been advocating for us achieving herd immunity, and people who have done bad calculations of these infection fatality risks and claimed that there's extremely low risk of death from COVID. So I think those have been harmful and slowed down effective policies.
Question from Peter Kessler. Peter, can you unmute and ask your question?
Hi, yes. I'm fascinated by your team's efforts. Great lecture. I just wanted to know, I'm a person that does support a great number of data scientists, in particular on the geospatial statistical side of modeling for not COVID at the moment, but you never know. And really just, have you guys looked at or can you describe if you have done, geospatial weighted regression, maybe Bayesian statistics that are considering the demographics of urban density, those types of things, and consider location as part of the modeling, or ties location into it. That's really, I hope that explains my question well enough.
Yeah, absolutely. So it's something that I know there are a few other big COVID modeling groups who have been really doing that and using a lot of sort of detailed demographic and other sort of healthcare capacity distribution and mobility style data in models to really make very detailed parameterized models. We haven't been doing as much of that, and I'm sure from your field's point of view, there's probably more data and more sort of variables that might be available that we don't know about. The particular cases where we've been doing this is in some of the studies of evictions, though now we're taking these models for other applications. So for example, we built a reasonably detailed model of Philadelphia, which included all different zip codes and based on a lot of different demographic factors about those zip codes, sort of classified them into different types and looked at different levels of social distancing and interaction, sort of the whole network of interactions between those zip codes, and used it to run models in that sense to really show that you could reproduce a lot of disparities that we know exist, which you would miss if you ignored all that, and also showing how those can lead to kind of worse infection outcomes than if you just kind of considered an average. But I think there's a lot more to do there. And the people that I had listed on the slides that work at Northeastern University in Boston, they do much more work of that sort. And I certainly know for other infections, particularly ones that have a more environmental component, there's a lot of epidemiologists who try to do that. It's not my specialty.
Thanks much.
Thank you.
We have a question from Charles Norton coming to us from JPL. Charles, good to hear from you.
Hello, it's good to hear. It's good to see you again, Larry. Can you hear me? I think I found the space bar as a way to unmute.
Yes.
Well, thank you very much, Allison, for your very interesting talk. I really enjoyed it. As Larry mentioned, I'm from JPL, so not only am I from California, but from the Los Angeles area, where COVID has been exploding. And I think back at the beginning, we were doing so well here in California. And it just makes me wonder, does your modeling or the modeling in general account for human behavior? I'm really curious to understand what happened here. I mean, from what I've observed, it seems like people have been social distancing and people have been masking. And I just wonder, could there be changes in human behavior where in some sense a certain percentage of a population just gave up and that led to the spread? Or could it be that, 6 feet as a social distancing limit may have been underestimated, and or maybe people just have not, taken into an account how changes in human behavior might impact the spread, given that it's been literally a year since we've had to try to adjust to these changes.
Yeah, it's a really great question, and something that keeps me up at night, honestly, trying to explain all of these patterns. And I think LA is a really good example in the sense that it's very hard to exactly explain. In a very rudimentary sense, one way of looking at this, which is one way we have been looking at this is like, what amount of these changes can you explain just by looking at like mobility data that tracks how often people are moving around or not. And like, you can't explain all of this by that. So then the questions are like, what are the other things going on in terms of like children dynamics as they're not tracked in mobility data, or things that people do in households, like, whether it's their household or someone else. It's hard to answer this. I sort of two hypotheses, which I think I kind of came up in the talk, is that like, I don't think, well, okay, in terms of the behavioral aspect, I think this is something that we really need a better like sort of social science of like, how do people react? How do people behaviorally react and change their behavior in response to like actual risk, perceived risk, duration of these interventions? I mean, those are all great questions. I think we have the answers to that. But just in terms of like what can be driving it in places like LA, I mean, I think, I don't think there's so many people are necessarily like intentionally giving up. I think it's just that there are many populations that are just not able to effectively socially distance. I mean, we have so many people still working and really like not receiving sort of the, financial and governmental support needed to protect themselves. So I think like a lot of that is going on, and it's amplified in certain populations. And, many people just can't reduce household spread, and that's also worse in some populations. So I think those are important factors, but it's a big question mark.
So I think Dick Garwin still has his hand up. Dick, do you have a follow-on or another question? Or is your hand is up from before? You have to unmic. Oh, okay. Then David Rabinowitz. David, do you have a question?
Yes, I appreciate the talk, and I'm just wondering, I assume you use Monte Carlo simulations to evaluate your stochastic models, and I'm wondering how much variation you see over the universe of simulation results, and just how meaningful the predictions are going to be.
Yeah, I mean, you see lots of variation, and usually we try to show that with like sort of some windows of likely outcomes. And this depends a lot on the particular question, but in general, overall trends, especially when you get to levels where like tens of thousands of people are infected, like are robust, but like the timings of things and the exact number of people infected, all of that has huge uncertainty bars on it. And when you get to the stage of infection, like sort of at the beginning or closer to the end of epidemics, where there's lower numbers and it's really being sustained by sort of rare jumps between like different subpopulations where like it's close to being locally eliminated, then that becomes very hard to predict. And based on the earlier question talking about evolution, which is like a very rare and stochastic event in models where you try to include like the generation and selection of new strains, there's even more variability. And then you can really only predict like likelihoods of one outcome versus a totally different outcome. Like it becomes very difficult in that sense.
We have a question from Michael Moore, and then we'll have one from Michael Hell, and then I have a few coming in on another line.
Hey, Allison, thanks very much for everything. This is a great talk. My question was about contact tracing. You showed a chart where you had a surge from, I think it was exercise classes, and then they traced the folks and it died out at the end. I haven't heard much or seen much on contact tracing, either nationally or locally. And I wonder, number one, do you model it on the scale that we have this infection going on, or is that possible? Or is there an effect that could be made by doing more of that? Anyway, thank you very much.
Yeah, so there's some really nice models out there about when contact tracing can be very effective. And there's certainly many places in the US are doing contact tracing, and there are some nice studies of the results from that, whether it's, like that dance class that I showed, like other sort of particular clusters that happened where they really mapped out how it happened and to whom, or studies where from contact tracing, they try to estimate how much transmission happens in different settings. But contact tracing works best when the epidemic is small, when you can really, like, there's some chance that you can actually trace back where, like, who you were infected from and find everyone that got infected. And like, the problem is that, like, we just let things get out of hand here. It's very difficult to do like effective contact tracing at the level of the epidemic that, like, we're experiencing, in the US and many parts of the world. Like, it can help, but it's just extremely resource intensive. We're probably not going to learn who got infected by whom, and contact tracing requires like really fast detection and notification. And that's just been a persistent problem. Like, you need to, like, feel sick, go to the clinic that day, get a test, and be notified a few hours later. And that gets sent to contact tracers, who then call you in your contacts like that day or the next day. Like, that's what you need for a disease like COVID for contact tracing to work. And we haven't been achieving that.
Question from Mike Houghton.
Yes, thank you, Larry. And I agree with all the other people that, Allison, you have a very good lecture here. I appreciate it very much. My question concerns one human body. From your starting remarks, it sounded like just like one COVID-19 particle that gets into the body can infect the whole body. And I heard that it takes about 1,000 particles to get into the human body in order for the body to be infected. Can you address that? And I have a comment concerning the LA behavior question that came up earlier that regards this same point.
Right. So, okay, I will say that the quantity that you're talking about, it's often called the inoculum size in this field. And in general, we don't know it. But important thing to say is, like, what exactly does it mean? So this is a very stochastic process, like this event of someone getting infected. And just because someone says, like, you need 1,000 particles of something to get infected, that doesn't mean like all 1,000 go and start infection in your body. It might mean that it's such a stochastic event with such a high extinction probability that the chance of success is only 1 in 1,000. So, you need to have 1,000 to have, like, greater than 50% chance of success. So often it can mean something like that, but really only one makes it through. This is hard to study for COVID, but for some other infections like HIV, for example, where it's so genetically diverse, like, we can really track like how many viruses sort of started the infection that ends up seeding a person. And, in the example of HIV, you can often trace it back sort of to a single virus lineage that's responsible for the whole infection. We can't do that type of analysis for COVID. So there's other ways to do it, but we just don't know. I mean, in this story, don't take that as like, my best estimate of how many virus particles were involved. But I think that, it's a chance event, and it's totally possible that only one kind of made it through and succeeded, even if many more tried.
So what I was thinking is that in a given area, like in the LA area, it might take some time to build up in many individuals so that they have a critical level of infected particles in their body before they really become infected. So this happens over time, so that at first the LA area seemed like a pretty good, very clean, but in fact, during that time, individuals were building up a critical level. And when they hit that critical level, then a lot of infections started to occur, and it seems like behavior changed when it really didn't. So I'm thinking that might be part of the game here.
So that explanation doesn't really work for COVID, because we do know these time scales, and the time scale of what happens in the body is just a lot shorter than like the whole sort of these trends you're sort of observing in LA. So when someone gets infected, obviously the virus starts at really low levels, and for some amount of time it's so low we can't detect it. That amount of time, is variable, could be between 2 and 10 days. Once it really starts going and replicating, it's very quick that it gets to the peak levels. So within, a day or two, so we know this because people that, we've tracked caught them kind of getting infected and sampled them every day this whole time. And this is a very repeatable pattern. Infection levels get very high within 2 days, and then they decay over the course of around a week. And then they're sort of undetectable again around then in most people. So this is not something that builds up over the course of months in people. This is like what we call an acute infection, meaning in most people, it's a very short time scale for this whole process.
Okay, thank you very much.
No problem.
I think we'll take a last question from the Q&A and the YouTube, and then there's just a couple more. So Kristen, you want me to read or can you ask your question?
Hi, I'll ask it. Great lecture. I've really enjoyed it. I have my telephone loaded with one of these tracing apps, and I'm wondering how effective they are. Have you seen any impact from the use of those in your studies?
Yeah, it's a great question. It's not something I've looked at sort of like evaluating them, now that they're implemented based on how many people are using them and how they work. I don't know. It's a great question. I'm not actually sure who is looking at that. I'm sure someone is. Feel free to shoot me an email. I can ask around and see if anyone knows, but I'm not sure. I know the many modeling studies that sort of considered scenarios for how effective they could be, but I don't know any that have looked now that they're started, if we can see the effect. It's a great question.
Yeah, I think one comment to the question before about about how much virus is required to cause an infection. Obviously, can vary for the individual. We do have immune systems. The immune systems do work. They don't work very perfectly and can't always recognize the virus as well as we like. But also there's person-to-person variability. I mean, huge variability. So
yeah, absolutely.
There's not really one answer.
Yeah, there's probably not one answer. That's true. Lots of physiological factors influence and likely influence your susceptibility to COVID. And we don't know what most of them are.
I have a question about the various methods to decide on when people are infected. Obviously, if they die, you can, to some extent, attribute their death to COVID. Although I think there's some inaccuracy in general, because they're not necessarily doing viral tests. And even if they are, just because you have the virus doesn't mean you didn't die of some comorbidity. But how robust are the models that you're using to the variation in the underlying inaccuracies and differences in the way we test for whether a person's affected, whether they've got antibodies, whether it's RNA, whether it's this test or that test, because the tests aren't all equally accurate. And when you compare the models to reality, to what extent are the variation and inaccuracies in the data that you have about reality problematic for your models for testing?
Yeah, absolutely. I mean, this is another source of uncertainty. There's uncertainty in parameters you put in when you're trying to fit to data. There's uncertainty in what exactly the data represents, like how you translate this thing I observed is like this thing in the model. Yeah, I mean, these are just extra sources of uncertainty. I think they've been narrowed down over time, but there's still issues. I mean, I would just personally say, like, the death issue, I think, is something that's sort of overstated a lot. I don't think, the process of attributing causes of death is actually relatively clear. Like, doctors are good at knowing like what killed someone. It's not so wishy-washy as people think. But certainly there are many places around the world where there are issues with death reporting. So this is why just a lot of uncertainty in terms of like how many people have already been infected by COVID. How does that vary across populations? Like, yeah, I mean, no matter how you spin it, this is certainly a problem that leads to uncertainty in parameter estimates and future predictions.
Is there any attempt, I guess, to quantify the range in which of the inaccuracy to see how it really, what inaccuracies are really going to make things terribly unpredictable, and what inaccuracies are tolerated by your models?
Yeah, I mean, and this is something that's like most relevant for people that are really trying to make like very accurate forecasts. I mean, most of what we're doing is what like me personally doing is like not sort of so tied to being able to like tell someone this number on this day. It's more of like showing, you know, these effects and can happen. But usually, you know, ideally, if someone is doing a good job with that, like it should be reported in like intervals, like on all of the type of predictions that are made. So, I mean, in some COVID papers and in some of these sort of app-based forecasters, like you will see that very clearly, like those windows that are around everything, they take that into account. It should be taking all of these things into account. Sometimes there are certain things that are assumed, and so it's not uncertainty of them, and other things are. The other ways, like many sort of different philosophies to doing this, whether it's like you just show an error bar versus like another way, sort of more common in like scenario modeling, is to really just consider like a few different discrete values of this parameter. Like let's say we don't know the duration of immunity, of natural immunity to COVID. So maybe I'm going to consider a scenario when it's 6 months, when it's a year, and when it's 2 years. And I'm going to show that you can get totally different outcomes in terms of like what the epidemic path will be given our uncertainty in that. So, you know, it's too big to even just show a window, the shading, everything would be shaded. So you just show that these scenarios, very different things can happen. And then the take home point is just like, you know, we can't say, there's no point trying to make a prediction for 2 years from now because we just don't know. So sometimes it's really that extreme, like because of everything that you're saying.
So a kind of related question is, there's a lot of large scale studies going on right now of vaccines, and to what extent are those studies attempting to employ other tests? Like I know some of the studies didn't actually test for the virus, and I don't know that any of them are testing for antibodies and the virus. And I don't know that they're comparing the different tests that are available to see what is the inter-test variability and try to get a handle on what the actual accuracy in practice is. So do you know if there's much of an effort to really systematize this into the studies that are going on to leverage the, you know, it's a very careful study. We have all these people captive really in the study. We could do all these things, but it's not necessarily germane to the endpoint of the study.
Yeah, so I guess I'm not completely clear. Do you mean by testing, do you mean testing people like before they get vaccinated, or do you mean like testing them after, like if they're in a vaccine trial, or just the routine testing that would happen
have
anyways?
An ongoing clinical trial. You decide that your endpoint is whether or not the people in the study develop symptoms.
Mm-hmm.
So you have a clear set of criteria for the symptoms you're talking about, but you don't bother testing for antibodies. You don't bother testing for the virus. So I think some of the first studies that were done, the 2 vaccines that are out, they didn't do tests for the virus or the antibodies. So I'm wondering if there's an impetus, some of the later studies, some of the ones like the J&J study, to get that information, because it's useful and important information to have, but it's not necessarily germane to the endpoint of the study.
Yeah, sure. So I'd say some of this is actually just because of the nature of how you have to design a clinical trial to like likely get approval. So they have to kind of pick like a primary endpoint and say, this is the thing that we'll use to decide. It doesn't mean they weren't measuring other things necessarily, right? And in many of these studies, like now, you know, they're, you know, going back and testing samples, and they are measuring some of those things. And there are many other like post-approval studies. For example, I know with some of the big healthcare systems here, everyone who they give the vaccine to is kind of enrolled in a study they have, in which they do like many more tests. But it's sort of unfortunate that in some ways that they kind of had to just pick one thing to say was their outcome in the trial. But yeah, I think we will gain, now that they're approved, they can kind of, it's less risky to measure all these other things. So yeah, I think we'll learn a lot from that.
Well, I think we're out of questions for the moment. And if we give people more time, they'll probably have more questions. So it was a very, I thought, very cogent, well-presented, excellent talk. And I think brought some clarity to modeling and viral transmission and the complexities of trying to make good policy decisions with all of the politics that's involved based on the best science and the best information science can offer. So I really appreciate your coming and giving this talk tonight, Allison.
Thank you so much for having me, and thanks everyone who's here for so many really great questions.
So thank you everybody for joining us. The recording of tonight's lecture will be available to everyone without charge on the PSW Science YouTube channel, the PSW Vimeo channel, and the PSW Science website. Please share the links with your friends and subscribe to the channel for notification on new postings. And if you're interested in the Society and you're interested in science, please do not hesitate to join. It's easy to apply for membership using the Join button on the homepage of the PSW website. Before you go, a few important closing notes. The next lecture will be in 2 weeks on February 5, 2021. The speaker will be Avi Loeb, distinguished professor at Harvard University. He will be speaking to us on the search for life elsewhere, not on Earth, and the likelihood of extraterrestrials, including ones smarter than we are. Additional lectures of the spring series will be posted in turn to the PSW website. Check there often for updates. You can see the dates of the upcoming lectures on this slide. And last but not least, please join me in thanking tonight's crew, James, Ann, and Robin, for producing tonight's event. And I will now adjourn the 2,433rd meeting of the Society. I thank everyone for joining us, and I wish you all a good evening, or whatever it is where you are, a good rest of the day. With that, the meeting is adjourned.